Hello.
Hello.
How
How are
are
you?
you?
Good.
I like the artwork.
Oh, thank you. Yeah, that got up there about midway in the spring quarter because I'm in the basement and it looks very dismal most of the time. So I
Needed some color.
requested some help. This one's mine. Can you tell?
I did not know that.
Isn't that nice? Are you?
Well, this one's mine.
Oh, that's nicer than mine. Good.
Art was my
Good.
first passion, even before science.
Yeah, I think I knew that. Okay. Carolyn, do you have any feelings about me recording this? Or you recording?
I was going to record it as long as that's okay with you, because I already had Jim Montgomery from Environmental wanted to check it out afterwards.
That would be cool.
Okay.
I would like to share. I see we're recording actually right now, right?
Yes.
Looks like it. Okay. That would be great. I was hoping to share with some folks in my lab that couldn't be there too.
And I can trim this beginning chit chat business off if
Thank you.
that would be helpful.
I will share. Can you see okay? No. Not
yet.
Do you want me to share yet?
It's up to you. I was gonna do a little intro once we hit 2 o'clock or shortly thereafter, but
yeah. So there's currently the book club, the anti-racism book club that's happening right now.
Oh, really?
Mm-hmm. The discussion of Kendi's book is ongoing.
So I scheduled this poorly, is what you're saying?
You scheduled in advance of everything else.
Yeah.
I thought that was an ironic conflict or tragic conflict.
Well, it just means there's enough of an audience for related topics that we can both get good attendance.
We can't hear you, Margaret, but I'm waving at you.
I wasn't even waving at you, I was waving at Sean.
Oh, we couldn't hear you that either.
I'll wave at you too.
Thanks. I haven't seen many of these faces in a while.
John, are you Movembering, or is this just a COVID thing?
Yeah, this is just a COVID thing.
Yeah.
We'll see. If it gets long enough, I'll probably keep it and then use it for cosplay or something. I don't know.
All right, they're pouring in now.
I'm going to share my slides, but I'm going to try to keep the chat function up. So if you ever have any questions throughout this, feel free to pop them in there, and then I will go into PowerPoint mode. All right, thumbs up. Are we good? Okay, great. All right, well, yeah, thanks again. I had a hard time naming this. I think what I should have called it are environmental pollutants, racism, and COVID, but really it's a weird combination of all of those thoughts. So I want to start with acknowledging the first is that DePaul in Chicago sits on the traditional lands of the Potawatomi Nation, and that there's still 75,000 tribal members living and working in Chicago. The next is that some of the funding for this initial study was coming from National Institutes of Environmental Health and Sciences out of NIH. Another is that I'm not really an expert on a lot of the environmental and structural factors that I'm talking about here. My training is in neuroscience, my PhD is in neuroscience. A little bit of endo, a little bit of immuno, and I'm just kind of purposeful research. And I say that to make these topics accessible to everybody, right? Everyone can approach these challenges from different perspectives, and perhaps that's probably beneficial. So there are lots of other experts out there, including some folks at DePaul. So there's a Center for Community Health Equity here. That is a great resource that I've learned from Dr. Mayo in that respect. And then finally, folks in my lab. So I'm going to show a little bit of data from our lab that Catherine Walker, Simone Rhodes, Debbie Lieberman, and all the other names that you see on here from bio, health, neuro, psych have been involved in. So the lab's general research interest is on brain development. And I think this image is useful. Can you see my cursor okay on this top sphere here? Great. So imagine this to be some eventual phenotype. So Waddington came up with this concept thinking about how cells developed, right? You're starting with kind of a basic precursor cell, and then there are different choices that the cells can make. And by choice, we just mean some influence is causing it to nudge one way or the other in its phenotype over time. And you can run down these different canals and wind up at this far extreme phenotype, or you can be bumped over to the right and wind up at this phenotype. And you can't go back up the hill. And it's harder to go across this way than it is to have just let gravity do the work. So what are these nudges that are bumping the phenotype one direction or the other? It could be some programmed effect in normal cell development, or it could be things like hormones or stress or diet or environmental contaminants. And we're broadly interested in thinking about the intersections of all of these. So my goals for today are to convince you of a couple things. One is to just get you familiar with the alphabet soup that is environmental contaminants. The next is to kind of demonstrate some of the molecular cellular effects of one example, and that's where the PCBs and our lab work comes in. The next is to introduce ideas of environmental racism and structural injustice. And some examples that I'll use for that are lead, air pollution, and racism, but there are lots others. Disturbingly easy to find examples of this. And then we'll want to consider this in regards to COVID, and then to pull you out of your depression and convince you that there's something else that you can do about this at the end of the game. Okay, so orientation to environmental contaminants. There are lots of different flavors, I guess you could say. So there are volatile organic compounds that are light enough to be in the air. Diesel exhaust particulates is an important one right now. There are heavy metals. Lead is one that we'll get to in a little bit. And then there's just this mix of persistent organic pollutants, including some pesticides, and PCBs are the example that I'll talk about. I'm also going to talk about per or polyfluoroalkyl substances. I think I said that right. That's a mouthful. PFAS is an easier way to refer to that. And the trick about these persistent pollutants is that they're stable. The persistence, that's what that means, that they don't degrade, which means that they have the chance to distribute. So the Arctic is a source where nobody's doing industrial work up there. It's not where people are producing persistent organic pollutants, but based on trade winds and water movement, that's where a lot of them wind up for one reason or the other. So they're distributed and they can bioaccumulate. So that's what this image is showing, that there are lots of sources of contaminants from people. They make their way into the water where small creatures can ingest them. A little bit bigger creatures ingest those, and up and up and up the food chain until you get to some really concentrated amounts, especially in fatty, top of the food chain systems. So this example here is an orca, and you'll see an image of that in a little bit too. So there's groups out of the UN that have had a convention about how to deal with these compounds. And so there's a link here. I should also have added that I've interspersed hyperlinks throughout this whole thing. So I'll think about how to make either the PowerPoint or PDF of that or some resources available at the end. But there's a whole like worldwide convention on how to deal with these. And it's giving out suggestions that nation states can follow. The US tends to like not do a lot of it. So there's lots of information available for you there. Let's think about how we're exposed to compounds. So this is a representation of our daily life, right? But it's also the source of a lot of this stuff. So fragrant, well, let's see, I'll start over here. So chemicals, parabens or phthalates. If you went into your shower and you picked up a shampoo bottle, you'd probably find something that said phthalate or paraben at the end. They're present in a lot of preservatives and artificial fragrances. There are flame retardants. So depending on where you purchased your couch, it was treated with a compound so that it didn't light on fire if you let a cigarette sit on it for a little while. These are diphenyl ethers and these PFAS that are used as flame retardants. They're kind of nasty. I'll get to that in a little bit. Pesticides and herbicides, DDT was what started this whole thing. It's a book called Silent Spring in 1960s, I want to say, really started people paying attention to the stuff that we were putting out into the world. Building materials include PCBs that I'll get to, lead and paint, industrial waste, cleaning solvents, plasticizers. So people have heard of bisphenol, BPA, because you've seen those stickers on water bottles that are BPA free. So compounds make plastic more amenable to use in our stuff, but they can also leach out and be problematic. Surface protectors, triclosan. So it's a long, messy list. What do they actually do? So some of these are endocrine disrupting compounds, EDCs, and I use this term because I'm a neuroendocrinologist, but I also don't always love it because it seems restricting. So all these compounds that I just listed can have endocrine active properties, meaning they can screw with the way that your hormone systems normally work, but they often do stuff beyond that. Kind of, I guess, universally, they do things beyond that. They're widely detected in our system. So BPA, PFAS, PBDEs, and PCBs, in pretty much every human sample that you take, whether it be serum or breast milk, you're gonna find detectable levels of these guys. I included the chemical structures to get the chemists on board here, but also because some of you might notice some structure-function relationships with these molecules. These guys are the structures for steroid hormones. So estrogen, estradiol is an estrogen. Glucocorticoids, that's cortisol, that's your stress, that's a stress hormone. Thyroid hormone is really important for growth and development. And you'll notice similar chemical structures there. So what that means is that your body has evolved to respond to your endogenous hormones, but these folks can fit into the spots on the receptors where your endogenous hormones normally would stick and either turn them on or turn them off, agonize or antagonize those receptors. So they can screw with how your normal hormone systems would be driving body function. And what that means is that EDCs can be associated with a huge range of disorders. So because your hormones regulate so many systems in your body, there can be a lot of disorders that are being made more prevalent by exposure to EDCs. So epidemiological and animal research have identified higher associations between higher concentrations of these compounds with these disorders. So let's just pick on one. And I'm picking on polychlorinated biphenyls, PCBs. So they're called polychlorinated biphenyls because there are lots of chlorines around biphenyl rings. So different structures, so they do different things. Some of them, the rings lie flat, some of them the rings can twist, but they are binding with a range of receptors, including hormone receptors. They can regulate neurotransmitter transporters, intracellular calcium, and that's really important as a secondary messenger in regulating cell function. Reactive oxygen species. So if you've heard of an antioxidant, these guys are oxidants. So what are they actually doing? They've been linked with, again, a huge range of disorders, messing with endocrine function. This is looking at fertility, This is looking at body weight regulation as a potential obesogen. And this is looking at diabetes, PCB, and increasing diabetes risk. They have been linked with immune dysfunction. So this is early life exposure and inflammatory systems, the production of a vaccine response. So how well your vaccines are able to work. Are these immunotoxic? And just kind of what they're doing to blood cells in general that could have immune active properties. They're linked to cardiovascular disease. So inflammation in your cardiovasculature related to atherosclerosis and other disorders. The most recent paper was linking it to early mortality as a result. And then finally, brain disorders. And there are a lot here, A, because that's what I'm interested in, and B, because the brain does seem to be a really vulnerable target to PCB exposure. So the research was initially looking at just IQ, like neural development, cognitive function. But then it's expanded and kind of focused in on ADHD-related outcomes, autism spectrum outcomes. So early life stuff, but also later life stuff. So this is looking at depressive symptoms in aging adults and Parkinson's disease. So lots of effects. If you're paying attention, you might notice that a lot of these disorders have something to do with inflammation. And so apologies to folks who know more about the immune system than I do, but I'm going to oversimplify inflammation to just get at an initial part of immune function. That's this innate defense against bad guys invading pathogens from the exterior world. And what these cells do is they can be macrophagec, they can start to chew up and eliminate the pathogens. They send out all sorts of cytokines and other signals that can gear up other cells into going into a similar response that can continue or can resolve. So inflammation seems really important in these whole body responses to exogenous environmental factors. And inflammation can happen in the brain. And when it does, it's bad. So there are lots of disorders that have developmental origins and neuroimmune function. I also have sex bias on here because there's this possibility, because we're talking about endocrine active compounds and because different creatures, right, different brains develop in response to different gonadal hormone concentrations. Some disorders are more common in males than in females, or vice versa. So the developmental ones, we're talking about autism, ADHD, schizophrenia. And later in life, we're talking about major depressive disorder, neurodegenerative disorders, and potentially substance abuse. And another point that I wanted to make here is this concept that's actually pretty common in immuno stuff, and that's that immune systems or processes can be primed. So it's exactly what it sounds like. There can be some early insult or process that then makes an additional hit exacerbated, or it's a more intense response than it would have been otherwise. And this is occurring over aging is what this image is showing you. So an aged brain has generally more primed microglia. And then in response to, say, infection or injury, that can go into this kind of positive feedback cycle. People have heard of the term cytokine storm as it relates to infection. It's almost a similar process that's here. So positive feedback meaning it just gets worse and worse and worse. It's like some injury turns on microglia, which then injures cells, which then turns on more microglia, etc. The other point that I wanted to make is that this can occur over development. So there's ideas of critical sensitive periods in development when the brain is on purpose really listening carefully to what's going on in its external environment, and therefore can pay attention to these environmental contaminants or whatever other stressor we're talking about, such that later in life they're hyper responsive to an additional challenge that can turn into a dysfunction of the system. So I'm going to show you data from 2 recent experiments that we've done, and that's all the hard data that I'm going to show you from our lab. The first is looking at microglia in vitro, and this is work that Catherine Walker just completed her master's on. And here we're taking brains from neonatal rat pups and then culturing them so that we're getting these mixed cultures of glial cells, so not neurons. We're talking about astrocytes and microglia generally. We would take off the astrocytes so that we're left with a pure microglial or pure-ish microglial culture. And then we expose them to PCBs in a dish. So these are cells that are alive. They're supported by the media that we give them to live in. But then they're exposed to PCBs, one particular PCB, PCB 153, for 4 hours, 3 different doses. And then we give them an additional challenge with lipopolysaccharide. And lipopolysaccharide is a component of bacterial cell wall, and it's normally would initiate an immune response, which is good, right? You want your cells want to know if there's bacteria present, but we can use it as an experimental model. That's pretty common. So this is a 2-hit model, right? We're asking if PCBs are priming the response to LPS. And here's just one piece of data from this experiment. So let me walk you through the graphs because there's a lot going on here. We've got females and males. On the y-axis is their expression of the gene that codes for the cytokine interleukin-1 beta. And on the x-axis are your 4 different groups within each sex. So here's the control that didn't get exposed to any PCB. Here's a little bit of PCB, 0.1 micromolar, 1 micromolar, and 10 micromolar of PCB. And then we'll start up here at the top. So these are animals that didn't get any LPS challenge. They just sat in PCBs for 8 hours. And you don't see any major effects of PCB. So these little dots are indicative of 1 plate, 1 sample, and your means are shown here. And there's no overall effect of PCBs just on their own. But if you give them LPS, so here, this axis is maxed out at 20. With LPS, we're making the cells cranky, so there's a lot of cytokine released here. Here's the response without any PCB. So this is a normal LPS response. They're sitting about, like, I don't know, 35, 50-ish. And when you give a little bit of PCB in advance of that, it's a much potentiated response. It's a really exacerbated response. This is in females. A higher dose in females, it's not as significant, but we were only able to get 3 samples there. So we need to do more work there. In males, the response is pronounced at 1 micromolar. So it's a slightly different response there. But in both of them, the PCBs are priming microglial activity. So they're making them hypersensitive. Oops, and that's showing you that. All right, so what about in vivo? So in vitro models are inherently simple, and that's purposeful, but our brains are not simple. So what's happening over the course of development in a whole brain system? So to get at this, animals, pregnant dams were fed PCBs on vanilla wafers. They were very happy with us. And then we studied their offspring over the course of development. The exposure that we're using is a mixture of R-chlors that are representative of what's found in fish in the Fox River just north of us in Wisconsin, and also what's found in schools. So these bars are showing you amounts of, let's see, so x-axis is showing you the different PCBs. Every one of those spikes is 1 PCB congener, 1 flavor of PCB. And then this is an R-chlor mix, and this is what's found in schools. So we're using a mix that's relatively representative of what's in our environment. And then animals were injected with lipopolysaccharide, that immune challenge, a couple hours prior to tissue collection. So what we're able to do is, again, look at if PCBs are priming an LPS response. What are they doing to the LPS response? So we've got animals that were exposed to PCBs compared to oil. We've got animals that were not were exposed to saline instead of LPS, and then some that were exposed to both. These studies have been published. Debbie was lead author on the study of the adolescent animals that we just published this year, and Catherine will be lead author on one that we're working on for the adult animals currently. All right, and here's what we have. So we used immunohistochemistry to get at this. So you're using antibodies directed at your protein of interest. In this case, it's IBA1. It's a calcium channel that is specific to macrophages. And so if we're in the brain, the macrophage that we think we're picking up is microglia. So this is kind of the gold standard in the field for how to label microglia in the brain in rats. And what you can see is that these cells take on different shapes. So there's a cell, and here's an extension. So this is normal up here. There's extensions kind of around the body. they're pretty, they're ramified, they're spread out, right? Because microglia, their job is to pay attention to what's happening in the environment. But as they get activated, they switch into a hyper-ramified state, and then they can turn reactive and sometimes even amoeboid. So what's happening there is they're resting, they're surveying their environment, and then they're gaining macrophage properties to do that. They get a little bit more rounded. So in the hypothalamus of adult animals, so again, I want to emphasize that this is a really low dose exposure. The animals were exposed in utero and then through lactation, right? Because the animals are exposed via breast milk, it concentrates in breast milk. But they were weaned at postnatal day 21. These are postnatal day 90-ish animals. So this is a couple months after their last acute PCB exposure. And here's what we see. So what we're looking at are the percentage of the cells that are hyper-ramified or reactive. So they're shifting from this super hairy morphology to a more reactive one. Again, a bunch of different groups, but you've got females and males. The total controls, they weren't exposed to either PCB or LPS. Just exposed to LPS, that's the hatches. And then any bars with pink are exposed to PCBs. And then there's the one group that was exposed to both PCBs and LPS. So if we're looking at the hyper-ramified cells, the percent that are at this hairy state, the percentage of microglia is in response to LPS is reduced, but only in PCB exposed animals. So nothing's going on in females. In males, LPS isn't really doing anything to this. It's a really low dose of LPS challenge that we used. In PCB animals exposed to just saline, no LPS challenge, this is where they are. So you give them LPS and that number drops, or that percentage drops. So if you compare this to what's happening in the reactive cells, you can see, again, nothing much is going on in females or in animals not challenged or not given PCBs, but in animals given PCBs, you give them LPS and the cells become more reactive. A greater percentage of them are in this morphology. And so what it looks like is that PCBs are kind of shifting the microglial response from a hyper-ramified to a reactive response. So this could potentially be a pro-inflammatory thing. We have a lot more to think about here, but I kind of just wanted to link in what we're doing with the immuno stuff, and you'll see how it connects the rest of the dots as we go. I'm going to pause. Are there any questions on any of that so far? I'm going to open my chat box and make sure I can see. Okay, so what about what's going on in Chicago? How does this relate to us? Well, this is an image you might recognize where we are. This is the Indiana Harbor Canal. Chicago's up here. So as you're driving, if you're going to Michigan, like I often go through, you go through a really smelly area of the highway, and that's where this is. So the bars here are different sampling sites of the sediment in the harbor. And the peak of the bar is the amount of PCB that's present. So it's definitely present in industrial corridors like this. This was taken in 2010. This image over here was from 2008, and this is sampling what's in the air. And so actually, you can search Chicago PCB air mixture, and it's now an experimental model. So they've sampled what's in Chicago. They've detected novel congeners that weren't even present when we used the PCBs industrially a long time ago. And so yeah, PCBs are in Chicago. If you pay attention to this map here, let's see how PCBs map on to other contaminants in Chicago. So here's Chicago's toxic burden. So this is data from the EPA. NRDC has kind of combined all of this using a bunch of different benchmarks of contamination, including water pollution, air pollution, proximity to hazardous waste sites, exposure to lead paint, vehicle traffic, and it's showing you a heat map here. So the red is more exposed to stuff, the blue is less exposed to stuff, and they've also grayed out these industrial corridors here. So that's what's going on in Chicago. If anybody knows the Chicago area, you might notice some patterns here that we should be paying attention to. And that's when we shift into the next phase of this conversation. And that is to introduce the concepts of structural violence that I first learned from Dr. DeMaio, but environmental racism. So here's that map of Chicago. Here's a map looking at household income. So red, lower income, blue, higher income. You can follow, so here's 55, go in this direction. There's that same pattern here. Okay, and then let's look at where folks are living. This was taken from New York Times using census data. Blue are Black Americans, orange Hispanic Americans, red Asian Americans, green white Americans. So we're paying attention to where these hot red spots are. And it kind of maps onto income, but it definitely maps onto racial groups. Down here and here. So we need to talk about this. Maybe this is just new information. Maybe we didn't know that this was a problem. Oh, no, it's been a problem for a while. So if you can tell, this is data from 1987. I mean, this is like early computer printouts, right? Looking at populations and their communities and their exposure to hazardous waste. So this is the percentage of the community that is of a minority population. So in communities where there are 0 hazardous waste sites, on average, 12% of that population is of community or is of minority status. 1 hazardous waste site, 1 hazardous landfill, and at least 1 or a large hazardous landfill. You can see that communities of color are far more exposed. And this is how this maps out into Chicago. So you'll notice those same patterns where these black dots are where the population is at least 50% Black or Hispanic. And then in the circles are numbers showing the number of waste sites or hazard sites. So there's definitely some relationships going on there. And I included this image because I just couldn't get over the title. So this is what racism smells like. When we think about environmental racism and structural injustices, we think about, so environment, I initially think about chemical exposure. So air pollution, hazardous waste sites, right? We just talked about that. But lead exposure, I think folks are aware that that's an issue in Chicago. Climate change risks and water contamination. By the end of this, I'd like you to also consider access to healthy food and healthcare and time to exercise as part of an environment, right? But also racism itself, the stress of racism as being a really critical feature of one's environment. So here's, this paper just came out like a couple months ago, and it's really pretty incredible. So they're taking data from NHANES, and this is this really incredible long-term study tracking US population. And they've sampled blood, and then they're analyzing the blood for a range of contaminants. And what you'll see, this is a heat map over here. So the color of the boxes indicates the level of the contaminant presence in these different groups compared to white women. So here's Mexican Americans, other Hispanic, non-Hispanic, Black, and other multiracial groups. So each of these little cells is one group. Within ages, so you can see 0 to 11 years, teenagers, middle-aged folks, and over 51 folks. And we're the major groups that I've highlighted here. There's even more in the actual paper, but we're talking about flame retardants, heavy metals, phthalates, PCBs, bisphenols, pesticides, and these perfluoral substances. So there's a lot to take in here, but I kind of wanted your eyes to just rest on the proportion of these cells that are yellow, orange, or red. And what that means is that communities of color are more exposed. If it's yellow, orange, or red, communities of color are more exposed than white women. I should say this was all in women. Then let's zoom in on one of these clusters, and I'm picking on PCBs again. 19 to 20, or yeah, 12 to 19, 20 to 50, and 51 years old. Let's focus just on this older group here. Mexican Americans and Hispanic folks, not as exposed, actually less exposed than white folks, but Black Americans significantly more exposed. So there's a lot to think about here. Here's another exposure. Here's lead, and zoom across these cells with your eyes. That's a lot of orange and yellow and red. Okay, so let's think about lead next. And I want to highlight the work that's being done here at DePaul. Dr. Jim Montgomery, Dr. Julia Lippert have been really active in sampling lead in Chicago area, and then communicating with the community about their active participation in protecting and advocating for their health in regards to lead exposure. But rest your eyes on this map. So again, heat map where hotter colors, red is more indicative of lead. And there's this really striking number here that over 3% of the kids in Chicago have elevated blood levels. So lead is not great for your brain. What I'm showing you here are images of dendrites on a neuron. So neurons are the cells in your brain. They're communicating to one another to make you do and think and stay alive. Control are those, this is a normal dendrite. So a dendrite is where the neurons are paying attention, getting incoming information. And all the little knobby knobs on here are called spines. And the more spines, the more landing spots there are for incoming connections with other cells. So we like to see nice spiny dendrites in kids, especially because they want to make connections, and that those connections are how they're learning. These are normal dendrites, nice and spiny. And these are dendrites exposed to lead. But what does that do in terms of neuronal activity? There's a lot going on here. These are called excitatory postsynaptic potentials. Essentially, it's you give a neuron some electricity because they communicate electrically, and all of a sudden you're going to see their activity increase. And so normal is the white, that's controls. So they're sitting here just kind of hanging out, not doing a ton. You give their incoming cell some input, and now all of a sudden they respond really well to that input. So this is the normal response. They're sitting around and then they respond to input really well. Again, lead exposure, not so much. This is like my stomach gets upset when I see this. Okay, so what about other contaminants where there might be disproportionate exposure? Let's talk about air pollution. So asthma is a major issue, especially in urban communities where they're exposed to a lot of air pollution. And what's happening here is that you're breathing in contaminants that induce an inflammatory reaction in your lungs. And you heard me talk about inflammation a little bit earlier. You saw an interleukin, so that's a cytokine. There are other interleukins that do different jobs. They're communicating with slightly different cells, but essentially you've got an immune response going on in your lungs. And that leads to swelling and inflammation and makes the airways tighter and less effective. So you've got these environmental contaminants coming in, but at the same time, you've got your brain dealing with all of life's stressors, and stress affects everything in your body. So stress normally is going to help you change, like if you needed to run away from a lion, you'd want to send your blood to different parts of your body. And so stress hormones can change how constricted or dilated your blood vessels are in different parts of your body. Chronic stress can screw with that. And so not only do some kiddos have environmental contaminants causing inflammation in their lungs, but they've got these stress responses on top of that. So a lot of this work has been done by Edith Chen. She's also in Chicago. And I'm kind of trying to highlight people who are doing this work, and a lot of them are in Chicago, which I think is really fantastic. So these are ratings of how well controlled a kiddo's asthma symptoms are. Kids reporting, and the parents are reporting, and then they've broken this down into socioeconomic status. So the parents, oh, sorry, I should also say, this is the parents' childhood socioeconomic status. So we're thinking about asthma in a kid, and the amount of money and status that the parents had is influencing how well their asthma is controlled. So you can see that parents coming from higher SES, their kiddos were a little bit more better, their asthma was better controlled. So what's going on? We can also see that this is interacting at the level of the interleukins, the cytokines, the cellular responses to asthma. So blue lines are high pollution and black lines are low pollution. So generally, let's just look at low chronic stress. So here you can see that kiddos that are exposed to high levels of pollution have higher counts of interleukins, these signals that are pro-inflammatory. There are more IgEs, greater eosinophils, cells that are responding to this. So in low chronic family stress levels, pollution is definitely pushing these asthma-related outcomes. But what about in high chronic stress? The high stress kind of in and of itself can push you in that direction. So we've got these interactions between stress and environment, chemical environments that are mixing together to induce chronic disease. Okay, so how does this tie into racism? We just saw stress. Racism is a chronic stressor. It is a psychological stressor. Just the perceptions of being discriminated against, being treated unfairly, induces a stress response in your body. So this study says, Karen Sabin, she's at Loyola. She's sampled women 50 to 75 years old. They're all from Chicago, and she recruited folks who were at risk of cardiovascular disease. And then she did an experiment with them. So they agreed to be guinea pigs here. She gave them all a stressor. She put them through what's called the Trier Social Stress Test, where they're asked to do a couple things, like do some mental math and say why they're important or why they're good at something. They're put through a social stress where they feel like they're being judged. It's kind of a classic model for a stress response. All right, and then we're looking at their inflammation in response to the stressor. So this is, again, another cytokine in their blood in response to the stressor. So x-axis is time, hour and a half, and levels of the cytokine IL-6 is on the y-axis. The blue lines are folks that are reporting lower levels of discrimination, and the red lines are folks that have reported higher levels of discrimination just in their everyday life. So this is so well studied that there are different standardized scales for reporting discrimination, and it's just their perceptions of being discriminated against. Not that those perceptions are real, but that's really all that matters in this case, is feeling like you're being treated unfairly. So discrimination is pro-inflammatory, and inflammation is not good for your body. All right, so let's put all this together and think about how it applies to COVID. We know what the risk factors are for COVID. We know that there are underlying diseases like respiratory disorders and cardiovascular disorders and diabetes. All these disorders are related to inflammation. So racism is a big part of this. Then I also wanted to think about how an additional environmental exposure could could exacerbate the severity of COVID illness. Okay, so these are directly relating air pollution with COVID-19 mortality. We don't have solid, well, this is as good as we can do right now in terms of understanding cause and effect. But what we're doing, what these researchers have done is measured particulate matter in the air across counties across the United States. So you can see all these different, again, it's another heat map where the red is greater levels of particulate matter. These are really tiny, like these exhaust particulates essentially. And then they're relating that on a county to county level with COVID-19 deaths. And overall that is pretty striking. So the red again is where more people are dying of COVID-19. So an increase of 1 microgram per cubic meter in particulate matter 2.5 is associated with 11% increase in the mortality rate. The other disturbing thing here is that you could switch out these outcomes, income, education, and percent of the community that's Black, with this particulate matter map, and you'd see a really similar pattern. So there's definitely, the community is definitely thinking about how air pollution could be making COVID-19 worse, and that burden is falling disproportionately on Black and brown folks. Now I want to introduce a whole nother compound that I kind of I mentioned earlier, and these are called PFAS. So I'm bringing this up because I think they really deserve attention right now and people aren't talking about them enough. There's a whole mess of them, kind of like polychlorinated biphenyls. There's a whole bunch of different compounds that have similar structures. They're per and polyfluoroalkyl substances. There are over 3,000 of them. The acronyms that you might see are PFOS, that's Scotchgard. That's the compound that people used to put on couches to keep it from burning and getting stained. PFOA, that's Teflon, that's your nonstick pan. These aren't being used anymore, these have been phased out. but they're replacing with compounds that are very chemically similar and probably do the same stuff. In fact, there's some data suggesting that they're just as bad, if not worse, than the original components. So what's going on with PFAS? Well, we're treating our materials, we're packaging food in them, they're coming out of industrial products, and they're making their way into water. And so our water systems are really contaminated. So PFAS are not great for your immune system. This is Dr. Philip Grandjean. He's been thinking about this for decades. This was a study done in, I want to say this was 2012, looking at early life exposure to PFAS and a kid's response to the Tdap vaccine. So tetanus and diphtheria, it's a standard vaccine that you get as a kiddo. And they've linked amount of PFOS, in this case, in the serum at 5 years of age to the antibody response to the vaccine. Right, so the way vaccines work is you give a challenge essentially, and then your immune system should be kicking out antibodies so that if you ever came into contact with the antigen later in your life, your antibodies would be able to react more quickly than they would have if they had to develop on their own. Right, so the vaccine is just kind of kick-starting a normal immune response to an antigen. So what they're doing is they're linking the perfluoral chemicals at age 5. So they've taken blood from the kids at 5 years of age, and then they measure their response to the antibody or to the antigen the vaccine later in life. So at 7 years of age. And what you can see here is that, so your x-axis is PFOS concentration. Your y-axis is the anti-diphtheria antibodies. So more antibody, the better the vaccine response. And as you can see, as the PFAS concentration increases, the lower the vaccine response. And this is really statistically significant. And sometimes it's like as one incremental increase in PFAS. So it's PFAS, let's see, I think they did it as PFAS doubles, you almost see a halving of the vaccine efficacy. They've also, so this was taken 7 years old kids. They've followed this up into 13-year-old kids, and the relationship has continued. And it's so bad that the CDC has put out a statement saying, yeah, PFAS exposure is probably making communities more vulnerable to COVID. These data have been collected by the Environmental Working Group, which I've linked to at the end here. It's a non-governmental agent, it's a nonprofit, but is essentially the watchdog that's pushing the EPA to do better. So here they're linking all these different communities. The blue dots are communities where the EPA has known that there's a problem, that there's a source of local contamination. And you can see that these bars are showing how much PFAS is present in tap water. So known sources have more, but even in locations, including Chicago, where there's no known source of contamination locally, you still see pretty high levels of PFAS. This is the EPA's limit, 70 parts per trillion, and scientists are arguing that it should be significantly lower than that. So a whole bunch of links to what we know about that here. And then I just kind of want to nudge in here that this is, my talk is definitely turning into more advocacy than just pure science. And I just want to say that science is always somewhat political, right? People don't operate in a vacuum. And in this case, there's a lot going on related to this. So Dr. Grandjean, you saw their face earlier, has recently published, or is trying to publicize the fact that the producers of PFAS have known about this for a long time. It's kind of like tobacco scandal or big tobacco, where the producers of Teflon and Scotchgard were communicating with each other even in the 70s and had known that it was an issue. So there was a lawsuit against them, and this is a snapshot of that initial legal challenge. Science Friday had a really cool episode with the lawyer who was pushing the lawsuit. Folks are working on this actively right now. So this is Dr. Linda Birnbaum. She used to be the head of NIEHS, Environmental Health Sciences, environmental health and safety. And she recently retired because she felt like she couldn't say enough about PFAS, that it's an issue, and she kind of stepped down, kind of retired, was kind of nudged out because she wouldn't stop talking about them. And the argument from from toxicologists and environmental sciences is that you have to manage PFAS as a class, right? At the moment, they're registered on a one compound by one compound basis, but there are 30,000 of them, and they're doing the same stuff. So you've got to deal with them with the potential for additive effects or even synergistic effects, where you can't just have the cutoff be relevant to one chemical. They need to be added up. Okay, so that was a lot. What are we going to do? I would encourage you to do a couple things. One is to advocate with the EPA. The EPA has kind of been destroyed by our current administration. Hopefully it will be restored shortly. But they've not done a great job thinking about PFAS. And we need to stay up on our next administration to ensure that the people that are appointing to the EPA are going to act on this. Another major source that the EPA needs to deal with are mixtures that are used in fracking. There's just a hot mess of compounds that are in fracking solutions. So that's one thing. Stay active in talking to your representatives, emailing the EPA, bringing attention to this. The next is to vote with your wallet. So buy products that are lower in some of these compounds that we're thinking of. I want to highlight that Black women especially are exposed to some of these nasty compounds in personal products because of the injustice of beauty standards, right? So there are folks that are actively thinking about this right now. So use your resources. The Environmental Working Group has put out a really extensive list of compounds that are in all these different products. So they've got a whole bunch of products that you can search for. Like if you've got a lotion, you can enter in that lotion's name into the site and see what's in it, what they're concerned about, what they're not concerned about. The other just easy tip that I would give you is to just look at the back, and what you're going to see in your personal products are things like parabens. That's paraben, that's paraben. If it says fragrance, oftentimes these are phthalates, but fragrances are protected legally for some reason, so they don't have to disclose what the fragrance is. So unless they're saying that it's actually, unless they give you the source of the fragrance, it's something to raise an eyebrow at. So vote with your wallet. The more that we purchase, if you have the privilege of being able to make potentially a more expensive purchase, you can purchase things that are free from this stuff. The market will follow, and it will make things everything more accessible to people who don't have that privilege. The next is to work with your communities. If this is upsetting you, don't just go start a new group. Join one that's already been established, ideally by the people who live in the communities that are most affected. So I highlighted some that are really involved in Chicago, especially the southwest or southeast area of Chicago. And there's links to that at the bottom. And then finally, I hope you can see that you can approach these problems from any discipline, right? Chemists can think about how to design compounds that are less nasty. They can think about how to deal with pulling the compounds from the current environment. Public health people can think about communicating and ways to reduce risks. Epidemiologists need to think about what's going, can establish the disease connection. Neuroscientists, endocrinologists, toxicologists, immunologists, everybody can get on this. So if you like research, you can use your method of choice on this problem. The second to last one is be anti-racist, right? Racism is bad. So you can protect other people's health by trying to reduce the harm on them. Last one on this page is to don't despair, don't give up. Don't feel like you're fated for a life of illness and disease, because the stress of that is just as bad on your body. So think about this as a challenge that you can overcome, and you can use these tools that we're talking about together. And then finally, keep reading. There's lots to learn. I can share some of these links. I can copy them into maybe I'll do that. Once I'm done sharing my screen, I'll copy these links into the chat box. And just stay on it, stay engaged as much as you can. And with that, I'm happy to take questions. I'm going to stop sharing and so I can see your faces. Okay, thanks. So any questions?
I have a question actually.
Yeah.
So my question is more related to the first portion of your talk, PCB, and NIH is really kind of encouraging research that be inclusive in terms of include animals from both sexes, male and female, and which you did for your studies. And I just found that from the in vitro study versus the in vivo study, it seems like the sex difference are different, right? Like in your in vitro study, both male and female cells responded similarly to the PCB treatment. But in your in vivo study, it looks like male's response was more statistically significant, but not female. So is there any reason? Do you have any?
Oh, just about 1,000. Yeah, no, it's something that we're actively thinking about. And I'm really glad it looks like that Dr. Jerome noticed the same thing, that sex differences are real, right? So our brains, our body develop with different chromosome complements, right? Some cells have X's and some cells have Y's, but they also develop in response to different gonadal hormones. And because gonadal hormones regulate the function of almost every system and every cell in your body, they're bound to be differential susceptibilities and vulnerabilities and responsivities to endocrine active compounds. So what's going on in terms of our data? Why is it that females are actually more sensitive in vitro studies to the lower dose, right, than males, but in vivo, after that long developmental exposure, the males are more sensitive? It could be several things. It could be methodological. It could be that those in vitro systems, the cells are no longer in their normal endocrine milieu, right? They're not responding to the circulating hormones that are going to be modifying their responses. But those cells in vitro were perinatal, right? They're from neonate animals, and those brains haven't matured in a way that could make them differentially sensitive. And so the in vivo data that I showed you were in adult animals that have gone through their life's exposure to different gonadal hormones. And I should also have said that I didn't show you in vivo data from P1 and P40 animals, from neonatal and adolescent animals. And at P1, the response looks really similar to that of the in vitro study, but P40, both males and females are responding, and the responses are a little bit more of a main effective PCB pushing things, whereas in adulthood, it's more like the PCBs are modifying the LPS response. So my takeaway, thank you, you're like a plug, Jing Jing. My takeaway is that people who are studying toxicology need to get beyond the adult male model. You need to pay attention to sex, and you need to pay attention to developmental stage. And I should also add that our in vivo data, it depends on the brain region, right? So it's just not simple. We just still have a lot of work to do.
Were the animals cycled? Did you collect the tissue at a particular estrogen cycle stage?
The treatments. So the treatments, the PCB treatments were given while they were in utero, so that was a long-term exposure. The LPS challenge, we have some data that was, I don't remember exactly what we did. So a lot of our molecular data, we did track, and I gave the LPS challenge on their peak estrogen, progesterone stage. So Dr. Kipp knows that I'm talking about their nucleated day of their estrous cycle. For the IHC data that I showed you, we tracked it, but we didn't actually dictate. We didn't give the LPS injection on a specific day, but we just tracked it. So I've yet to finish that analysis to see if their stage of estrous cycle had anything to do with it. But I do want to say that for the community, there's lots of data demonstrating that even though there might be effects of estrous cycle on some outcome, it's still worth looking at females, even if you're not going to control for it. The variability that's induced by the stage of the estrous cycle is less than variability between animal for other reasons. So that's a long-winded answer to your question. And the answer is like kinda.
Yeah.
Can I ask a quick question?
Yeah.
I don't know if sorry, Jing Jing, did I cut you off?
Oh, no, go ahead. ahead. Okay.
Just with the internet, the international component, sometimes some of these things are illegal in the United States, but they're used either legally or sometimes even illegally and produced illegally in other countries. So with the Teflon, there's a movie called Dark Water. It's a big Hollywood movie. Do you know if that's, you mentioned that was now the production stopped in the US. Do you know if that's been banned globally, or is that?
I don't think it is. I think the fact that production's been phased out in the US was consumer pressure on the producers as opposed to actual regulation. And so it's going to really depend on the communities and who's paying attention. But with you raise a really good point that what I just described as US, it was all focused on the US. Different communities are going to have different exposures, especially in developing nations. So, and a lot of we send all of our electrical junk to landfills in other countries, right? There are communities that live near like computer waste sites that are just exposed to just disturbingly high levels of heavy metal contamination. In terms of regulations of PFAS and PFOAs, those were described in the Stockholm Convention through the UN. So I think their use has been reduced in most developed nations, but I don't know about legally what's going on in other countries. It's a really good thought. Thank you. I see a question from Dr. Jerome here. So I wanted to know if the male-female mice tend to react differently in the same way that male and female humans do. So is there convention that you can assume if male mice are more reactive to PCBs, will male humans do the same? That's a really fair question. And that's really assessing, like, are these rat models fair? Are they good ways to ask these questions? Male rats, just like male humans, are exposed to more testosterone than are the females. And conversely, female rats, just like female humans, are exposed to more estrogens, just like humans. We know a lot about the similarities between those systems. And so I'd say most of the time, yes. There is one factor that differs between rodents and males, and rodents and humans, and that's the timing of their sexual differentiation, at least of their brains. And the fact that in rodents, just to screw with you, the way that the males are masculinized is actually because they have local production of estrogen in their brain. So the way that gonadal hormones work is that testosterone is, I don't have it on there. So if you think back to those structures that I showed of steroid hormones, they're all based on cholesterol. And the cholesterol is modified. Little bits are snipped off here and there to make the different hormone structures. And the only way that you can make estradiol is to first make testosterone. And then you do one last snip, and then all of a sudden you've got estradiol. And there are enzymes that are produced in your brain that can do that. And the way that male rodents are masculinized is by the local aromatase activity right around day of birth. So that was a long-winded answer to Dr. Drum's question. So I'd say most of the time, yeah, I expect that if you do detect sex differences in rodent models, you're likely to detect them in human models. The mechanisms by that might differ, but I think we need to continue to pay attention to sex in humans because of all the sex differences in diseases that we see, right? Cardiovascular disease, way more common in males than females. Alzheimer's disease, way more common in females than males. CVDs, males, Alzheimer's, females. So I hope that answers your question. She said, yeah, okay, good, thank you.
I did have one quick question for you.
Yeah.
So I'm very neuron centric, and I kind of have this blind spot when it comes to glia. So I'm hoping you can educate me a little bit about this. So in your in vitro studies, you had seen that there was a change in the cytokine production. And in the in vivo studies, you had seen a change in the microglia morphology, right? Is that kind of the gist of it? Yeah. Do we know which way it goes? Is it the cytokines that influence the morphology of the microglia, or is it the microglia that produce the cytokines, or is it both, or neither?
That's such a fair question. I think the simple answer is that as microglia are getting ramped up and activated, they're going to shift morphology and produce cytokines. But they're also listening to additional cytokines So it's probably bidirectional. I should also say that the morphology is a really imperfect measure. It's kind of as good as we can get in tissues, like fixed tissues from animals that we just collected, using microscopy. Yeah, so they're not perfect. I'm sure a cell could produce cytokines prior to being in that reactive morphology. And the morphology might be more relevant to phagocytotic activity than cytokine production. So that's not a very complete answer, but that's as much as I can describe. Yeah.
Quick question about the PCB's function. So what kind of property do you expect that to help to carry out the effects that you saw? I mean, is it because is it estrogenic compound, or also the dioxin type compound or non-dioxin? I mean, what kind of property are we looking at right now?
Well, so the in vitro experiment that we did, that was with PCB 153, and those are non-coplanar, so it's not dioxin-like, it's not the acutely toxic stuff. 153 is anti-estrogenic. It's not estrogenic. So one thing that could be happening is it could be preventing the effect of an estrogen receptor from being anti-inflammatory, right? Because the simple dogma is that estrogen is generally anti-inflammatory, and so if you block that action, then it's going to allow inflammation to continue. What I suspect is it's a little bit of that, but PCBs have also been related to the enzymatic activity production of reactive oxygen species, and microglia use ROS to communicate with each other. It's a very pro-inflammatory process. So a little bit of that. And then I'm also guessing, and have proposed to do research on this, but I don't have the money to do it yet, that PCBs might be interacting with intracellular calcium receptors or channels that regulate microglial activity. So one of the challenges of these compounds is it's not a simple ligand receptor. It's not just one receptor that it's affecting. It can act through a lot of different endpoints, even within the same cell, and they're all going to interact with each other too. So ER, yeah, probably, but also probably ROS and potentially ryanodine receptors.